different antibiotic resistance, not only the vancomycin. So we was performing with the pure culture first. We know it that we can treat them, and after that we was doing a tool. You're taking sample from the patient, total samples, fecal samples can be even. And then you do the total DNA extraction. You do the treatment with the disc probes for the different antibiotics. You do the hybridization, that is about 1 hour. And then the flow cytometry takes about 15 minutes, and you can see something like that, and you can see the patient have presence of antibiotic-resistant genes. You're not going to give this antibiotic. You select the antibiotic that is more appropriate. So all this process can take less than 1 day. If you do the classical way, it will take you a week. In this way, patient coming to the hospital in the morning, in the evening, you already know what kind of antibiotics you can apply and how you can apply it in order to be effective. Can we talk about postbiotic a little bit, then have a little bit of this? Let's have a discussion and put the postbiotic in the after lunches, okay?
Yes, yes.
Because they informed me that I need to go for another meeting at 12:30. So let's do the discussion, and postbiotic we can do it in the afternoon session.
Yeah, no problem.
So what's your curiosity now about this part? Do you think probiotic could be really a good idea when we need to be careful? In my opinion, probiotic can have a very good application. I mean, they show it, like almost 1 century, we apply them in one or another form. The problem in the beginning, we don't know too much about them, and everything look much more easy. In these days, we go deeper and deeper, and we realize we need more and more study. You remember, there's a story from the old Greek time. Socrates, the philosopher, he was used to have his lecture with his students on the beach. Well, not too bad idea, but Greece is the peninsula, the coast is nearby, and one day the students, they asked Socrates, but you know everything, every time we ask you something, you have the solution, you know everything. He was taking one stick and draw one circle on the sand, circle, and say to the students, listen, this circle representing your knowledge. And here draw another bigger circle around and say, this bigger circle representing my knowledge. See, I know a little bit more than you. But now think about something else. This line around the small circle, how long is it? Representing things that you don't know. And now think about the long line around my circle, representing things that I don't know. So you see actually the beauty of the science. You go and discover more, and you realize in fact that it's more things to be discovered. And it's actually making it interesting, because you're not satisfied what you already discovered, what you already know. You want to know more, and more, and more, and more. So this is actually the interesting same thing with the probiotic. In the beginning we don't know too much about them, but we don't know what we was not even imagine what we don't know. So with going more deeper into the area of the probiotics, we realize that it's actually the unknown is much more bigger. And this is actually making space for everyone to work in the area and to try to discover these answers. So I know someone also already several times mentioned that work with the probiotics. And yeah, so what is your challenge in the lab normally? Feel free to share what is the problems, what do you think need to be done. You have some ideas. You see, in the beginning people was thinking that the fermented food products could be a good option. Even was several fermented food products was marketed as a probiotic in the supermarkets, because the word was selling almost everything. But then they realized the regulations and main regulation bodies, they say you cannot claim as a probiotic if you don't have sufficient clinical evidences for this. So they start to remove the name of these fermented food products and call them beneficial products, functional products, but cannot call them a probiotic because very often they don't adjust to the definition, because you need to know what strain do you have inside. You need to have the clinical evidences, how this strain is working, why it's working. And as well need to know the negative potential, negative effects. You don't want to put the probiotic products on the market that they have too many negative effects. Or if there are some, you need to aware the customers. So this probiotic is not good for you because you have diabetes, you have the high blood pressure, it's good for your mood. I mean, some beneficial metabolites like GABA, they're very beneficial for the nerve transmissions. But again, that was interesting because they could be, GABA is a very good option, but again, too much GABA is good, maybe not. Maybe needs to be a right dose. I mean, Paracelsus was telling that the poison, it's a dose dependent. Something that in the high dose maybe it's a poison, in low dose it's a beneficial. Homeopathic, it's the same thing. Some small amount of the metabolites, they can stimulate the immune system. So now the homeopathy is a probiotic? No, because it's not live microorganism, it's a metabolite. But again, can we maybe call the postbiotic homeopathic products? Maybe, why not? Because you have the metabolites, you provide some metabolites in a certain level, and you stimulate the immune system, you have the beneficial properties. When we go to the postbiotic story, you can realize that in fact several of the probiotics benefits, they are realized on the some metabolites produced by microorganisms. And these metabolites can be even provided without live cells. They don't need to be alive. They are good because they can be produced during the lifespan. And if you have the cells that is colonizing your gastrointestinal part, they can actually produce constantly these metabolites. But then is coming a problem. What about immunocompromised people? This microbiota there, it's a good or it's a problem. So maybe the application of some semi-purified or some even crude extract, but not a live cell, could be a good option. You see, the questions start coming and coming because when Mechnikov and Stam and Grigorov, they claimed that yogurt and Lactobacillus bulgaricus is good, was simple. There was not metabolomic, not genomic, no proteomic. It's not all this study, but in fact, the development of the technical advances in the science was helping to make a research and to answer the questions more complex and in shorter time to perform the experiments that will give the idea about the benefits, about the safety, about the danger of these microorganisms. No questions, no comments, more chocolates. I think someone wants to ask in the first row. It's a shy but wants.
No. So actually you discussed that bile reduces the adhesion property. So what could be the reason behind this?
Ox bile.
Yeah.
Well, this could be changed in the presence of ox bile. Most probably could be changed on the surface. Maybe change of the electrical charge. Maybe it's just simple interactions. Maybe it's addition of some other components is better. But again, think about biles where they are. They're more in the upper gut. No, right after the stomach. What's the role of the biles? They need to reduce the cholesterol. They need to perform in the metabolism. If you go further in the way on the gastrointestinal part, they're reducing. So in fact, this addition, low addition in the presence of bile will give the opportunity the probiotic to adhere to another part of the gastrointestinal part. Sometimes you don't want them to adhere to one to another part. You want them to adhere to the place they can perform their work. Because if they're producing vitamins in the mouth, the vitamin is not going to reach place down when it's supposed to be. So you want a probiotic to perform his action in the right place. Again, you can do it by encapsulation this way. You can keep it probiotic with specific capsules, so it can be resolved in the place you want probiotic to be acting. Or sometimes the natural interaction in our body was actually preventing the probiotic to be better in one or another place. Actually, you know, the nature was already doing the job. They created the human in the way. And sometimes we just need to be carefully touching this because it's a very delicate system. If you touch something that is making some changes, you maybe will regret later. You know, the same thing when the people in the beginning of 20th century, they was using radioactive water for some treatment because they observed that have some short-term benefits. But no one think about mutagenesis that is actually performed by this radioactive environment. There was several radioactive, mineral water in general could be a little bit higher radioactivity because it's coming from the deeper parts of the earth. And in some situation, they was recommending this water for some rheumatic or other diseases. To do the bad with this water, but then you need to put on the balance. What about the negative effect of this radioactivity? In the beginning of 20th century, they was not aware of this. This day we know much more better that radioactivity is not a good option. But again, every medicament, it don't exist the medication, the pharmaceutical product that is only beneficial. You helping the patient on one side, but you giving a problem on another side. So the benefits needs to be more than the damage. If we have disease, we don't have option. We need to take antibiotics. We will destroy our gastrointestinal microbiota, but this is coming the probiotics and fermented beneficial foods to restore the microbiota. Again, yesterday, I think it was yesterday, we discussed in the afternoon session about the fecal transport. So it's a way to restore the microbiota after Clostridium difficile infections. Because of the high level of antibiotic treatment, you're going to have this reduction of the normal microbiota. Clostridium survive and is taking over and performing the problem. Fecal transplant is one of the options. It's been applied for, actually been applied for a long time, but in the Western medicine they started recently to apply because it's just need to be sure that you have the donor with a healthy status. Because you don't want to introduce another disease. Poor person have Clostridium difficile, and now we're going to introduce Salmonella. Obviously, it's not a good idea. Any comments? Yeah?
Sir, do probiotics have any specific level of acidity tolerance, and this is totally dependent on what type
Sorry, it's too, come in the front.
I want to ask, do probiotics have any specific level of acidity tolerance, or it is totally dependent on what type of probiotic we are using?
If I understand well, it's about the acidity.
Acidity.
Yeah. Probiotics, they are bacterial culture. So obviously they will have their personal preferences toward pH. If you have lactobacilli, they are much more happy with the 3.5, about 4 pH. They can survive even a little bit lower, but enterococci was acidification of enterococci going to 4 point something, but they can have better survival in the lower pH. You see the growth condition and the survival, they are not in parallel, sometimes they're different things. Even Salmonella, you can kill it with pH 4, but important is when you have the probiotic, you need to be sure that the probiotic will survive the acid conditions, since he needs to go via the stomach. And in the stomach is like 2. Most of them, when they select, you do pre-selection, you look for this one that can survive. Because from the technological point of view, it's much more easy to give the pure bacterial culture. If you need to encapsulate, to incorporate, or something, or make a specific pharmaceutical formula to, it's giving more headache. More simple, it's more easy. But if you have a strain that have a very good properties, a very strong immune regulations, very strong antimicrobial properties, very strong production of neurotransmitter, etc., you need to, and he don't survive, you need to go to these additional methods and to be sure that you can deliver in the right place and in the right conditions. Because technically our body was built to protect. If you think about any bacteria that will come in our mouth, we don't want this bacteria to go down from the safety point of view. We have different enzymes in our mouth, we have saliva, we have beneficial microbes. There are more than 400 different species that will try their best to kill any new bacteria. Then things going down. In the stomach, very acid condition, they will try to kill anyone because nature wants to protect us. They made all these barriers in order to protect what's going in our gut. Then it's passing after the stomach, you have bile salts. They will try to kill as well. So you see, probiotic, it's a good guy, but nature was already create us to protect from any microbial invasion. So in this case, we need to select the strain that is a good survival from one side, or if we cannot survive, we need to help this strain, because we're now talking about the additional, not normal, this is extra thing. Probiotic is not something that is part of our body. We're putting these things. We're selecting one from our body that is already beneficial, and we're putting in another person. So we need to find a way how to protect them, to be ordered that they arrive alive in the place where we want them to perform. But then it's maybe one more philosophical point. If the nature, the evolution, create the humans in this way, why do we believe that we can change all these things in a week or 2 weeks? It's a little bit. Okay, if you believe in new religions, if the creation you by your way, how do you believe, it's done by this way, why we should change these things in such a short way? Who gives us the right? Well, the right, we want to help the people. And when someone is struggling, we want to be good with this person. So in fact, the probiotic is our wish, good intention to improve the quality of life of our friends. But again, it's need to be very careful, because if we mess with something that we don't understand, maybe we will regret later. So rather do it everything with the appropriate responsibility to be sure that we're not going to create a new Frankenstein. Maybe we can have a break now.
Yes, sir.
Everyone wants a break, right? Okay, please, actually I don't know, at 2 o'clock we need to be here or in the other room? Okay. Okay. Because Santos was mentioning to me something, but I was in one moment confused because, okay, I know we have a lecture,
yes,
but again it was where will be here, there, so here.
Yes.
Confirm it.
Yes,
Okay. So please, I will try to change now to the PDF, and I will send to team of Santos, and then you can distribute to everyone.